We'll go on to Taylor. Austin, if you could move to Michael's presentation.
All right. Hi, everybody. My name is Michael Stickley. I am a member of the US Youth Fly Fishing Team as well as TU Teens, which is what I want to come and talk to you guys about tonight. So I just want to talk a little bit about the 2023 Teen Summit as well as kind of what we do as the TU Teens. So, next slide, please. So TU Teens is a group of teens from across the country who are passionate about fishing and conservation and are learning how to become trout unlimited leaders within their community. TU Teens strives to promote a diverse community of passionate youth who foster a sense of stewardship towards America's fisheries. So this year's summit was based out of Georgetown Lake near Anaconda, Montana. We had teens from 15 states attending this summit. It was 5 days. It was from June 28th to July 3rd. And we had a lot of YLC sessions aimed at getting more youth involved in TU. We did a lot of fly tying, a lot of fishing, a lot of conservation, and we got to meet and hang out with other teens that are like us who are passionate about trout fishing and conservation. So our first conservation project that we looked at was the Berkeley Pit in Butte, Montana. So this was a former open pit copper mine that operated for 27 years. Um, it is 7,000 feet long, 5,600 feet wide, and 1,600 feet deep. The mine is filled with acidic water and toxic materials that kill any wildlife that come in contact with the water. And it's very acidic. It has a pH of 2.5. And the water from the pit gets treated to remove toxic materials and is discharged into Silverbow and Blacktail creeks. And we fished Silverbow Creek after we toured the pit, and you can definitely tell it has a lot of impacts from the toxic water that's been released into it because the fishing was just awful. So, all right. So our next conservation projects were beaver dam analogs on a tiny little creek called Smart Creek. Um, so beaver dam analogs are man-made structures that are used to mimic the form and function of real beaver dams, and they improve water quality by trapping sediment and slowing the water flow of the creek. And they increase pool habitat and cover for West Slope cutthroat trout, which helps them thrive in streams where they otherwise wouldn't. So these beaver dam analogs, what we do is we just go in and we put a bunch of posts in the bed of the creek, and then we pack willows, mud, rocks, any sort of debris that we can around that, and pretty much create a mock beaver dam. And if there's not already beavers in the system, Montana Fish and Wildlife will essentially stock beavers into the creek. And the beavers will either take up residence in the fake beaver dams or they will just disassemble it piece by piece and then use all those materials to make a new dam, which not great for us, but good for the beavers, I guess. So our first day of fishing was on Flint Creek. It's a small little outlet stream of Georgetown Lake, and it's low gradient, just a little meadow stream with predominantly brown trout, but also a few cutthroat brookies and rainbows mixed into it. So our next day fishing was on Rock Creek, and Rock Creek is widely regarded as one of the best destinations for fly fishing in Montana. And this was really fun because we got to fish in big groups and we fished with people that we just otherwise normally wouldn't fish with. Now, that being said, the water was up quite a bit and it made wading really, really hard for us. So a lot of us fell in, but we still had a fun time despite being really cold for the rest of the day. So, but yeah, we just spent a lot of time fishing with each other and bonding and just getting to learn techniques from across the country. We also caught a lot of whitefish too, which not everybody likes them, but at TU Teens, we really value our native fish, so we like them a lot. We also spent a lot of time doing leadership and team building workshops where we were just discussing how to get more youth involved in TU Teens and Trout Unlimited. So some of our ideas were teaching somebody new how to fish, whether it's somebody who goes to our school, a community member, a family member, really just anyone who's willing to learn. Um, We're also talking about establishing fishing clubs at our high schools across the country and taking TU teams items such as stickers or really whatever else we can get into local fly shops. But we also did a lot of we played a lot of volleyball. We played soccer. We hung out around the campfire, and we did a lot of fly tying. So, all right. And then we also just made a lot of friendships too. I think everybody who was involved in the summit, made new friends across the country, and they will definitely be lasting a lifetime. So, all right. So to apply to the Teen Summit, whether you're interested or if anybody you know is interested, you just have to fill out an application, and two recommendation letters as to why you should attend the summit. And this year we're not doing a summit due to some scheduling changes this year, but we are doing a excursion, which is pretty much just a 1 week camping and road trip style um, teen summit. So yeah, that's it. Any questions? Yeah, I have. So there's a kid, his name's Oliver. He lives up in Boulder, and I've fished with him a couple times since the Teen Summit. So and I think there's a kid from New Jersey who's coming out here this summer, so I think I'm gonna try to fish with him a little bit too. So all right. Anyone else? All right.
Thank you. Well done. So now, so now we're going to listen to a presentation from Emma, who is our scholarship award recipient. For those that don't know, that annually we get a scholarship to a CSU student of individuals. And at the end of their time with us, the three-year scholarship, they come and give us a presentation on this. So, let's go. We got my favorite here, and it's going to talk to us a little bit about fire and the impact on genetic diversity, right? All right. So, and that's as far as I can get with this. So, Gary,
Sounds good. Yeah, well, thanks everyone for coming. Thanks for the invite to talk about my research and also for the really generous scholarship, which has been a huge help for my graduate school career. So I'm going to be talking about a little bit of the work that I've been doing for my master's degree up at Colorado State University. Specifically, this aspect of my research is looking at the influences of major wildfires on genetic diversity and like spatial population structure of brook trout. So a little background about wildfire trends across the Western United States in general. So where a lot of data has shown that fires are increasing in frequency, so they're happening more often, and they're increasing in spatial extent across the Western US. And that's kind of what you see in this figure down there on the bottom left. And this was very apparent to residents of Colorado in the summer of 2020 when the Cameron Peak and East Troublesome fires both burned on the Front Range area near Rocky Mountain National Park. And these are the two spatially largest fires that in Colorado history, and they burned a combined total of more than 400,000 acres. And you can see the view of the Cameron Peak fire from Estes Park right there. So these were some pretty impactful fires for Colorado. And so events like this are expected to have pretty severe effects on headwater stream habitats. And so we see things like loss of vegetation, decreased soil infiltration capacity of water, which causes very drastic erosion. So the soil can't soak in the water, essentially. And this causes ash input to stream channels and severe flooding and debris flows. And some research that was done by my advisor and collaborators at CSU showed that there has been declines in trout abundance at severely burned sites following these fires. So if you see this figure here on the right, it shows higher trout abundance pre-fire, and then it dropped 1 year after those two fires. And then we saw population recovery happen the following year. So this shows that trout populations are affected by these fires. And so I want to talk a little bit about how these types of things are expected to affect genetic diversity. So whenever you have an event that reduces abundance or causes mortality within a population, this is expected to decrease genetic diversity because the subset of individuals that survive or recolonize to recover that population has a lower subset of genetic diversity, which causes lower genetic diversity in the recovered population. And low genetic diversity is a problem, especially for small and isolated populations, because it can lead to things like inbreeding depression, where individual fitness is reduced because of increased rates of homozygosity and things like that. And it can also lead to populations having lower adaptive potential because there are fewer genetic variants that can differently withstand certain environmental pressures and diseases and things like that. And specifically for restored populations of native trout, genetic diversity is a concern. And so if you look at this figure here down at the bottom, this shows on the far left of those two facets, genetically pure greenback cutthroat trout fry have much lower survival and growth rates compared to green lineage Colorado River cutthroat trout. And this is because of the severely low genetic diversity in greenback cutthroat trout. And so those three other ones are, I believe they're green lineage Colorado River cutthroat trout and hybrids with greenback cutthroat trout. So because of their higher genetic diversity, they have higher survival rates and growth rates of fry. And so things like wildfires that can impact trout abundance and potentially genetic diversity are a major concern, something to consider for native trout recovery. And so this kind of brings me to talking about the Poudre Headwaters Project, which is the largest reclamation project for greenback cutthroat trout in Colorado history. You can see the area, the shaded area there is the reclamation area for greenback cutthroat trout where they will be introduced. So the goal is to restore greenback cutthroat trout to a large stream network of 37 miles of stream habitat in the upper Cashflower Poudre River Basin near kind of upstream of Fort Collins. And this will come into play later in the presentation, but I wanted to set the stage for those who might not be familiar with Poudre Headwaters Project. So the objectives of my research were, first of all, to look at trends in trout genetic diversity before and after fire. So we've looked at abundance and seen that it decreases after fire, but no one has actually measured trout genetic diversity before and after fire to see if there were effects. And based off of that previous work that was done by other researchers, we hypothesized that genetic diversity will decrease in the year following fire, and then we'll see recovery 2 years following fire. And then my second objective was to examine spatial genetic structure of brook trout within the Poudre Headwaters Project area. What I mean by spatial genetic structure is it answers the question like, are there distinct genetic groups in this area? Or is it a genetically homogenized area where it's a single population where individuals are moving freely throughout that? And our hypothesis was that genetic structure exists in this area and that it was disrupted by fire, which reflects local extinction of severely affected areas and then colonization. So this is the study area that shows all of my sampling sites. So the black dots were sites where we sampled brook trout. And you can see the area that was expanded upon there is the reclamation area for the Poudre Headwaters Project. So we have a higher density of sampling sites within that area to look at spatial genetic structure. And you can see how these sites happen to kind of be half of them, luckily, maybe not luckily, but they were burned in this fire. And so it produces a unique opportunity for us to look at the effects of fire on trout populations where we have multi-year sampling sites, and an assortment of control sites that weren't burned, and then a good amount of sites that were burned by these two fires as well. So what we did at these sampling sites is we used backpack electrofishing to sample the brook trout, and we measured all the trout for total length to get an idea of the age structure of the populations. And for genetic samples, we took a little fin clip from the anal fin of the brook trout. And then you can dry these and store them in envelopes, and they're very good for a long time. So this is an effective way of taking genetic samples from fish. And so then I brought these samples back into the lab and genotyped them at 12 different genes. And so if we kind of remember from biology class, if you have different genes, all the individuals have the same genes, but they might have different versions of those genes. And so those are the different alleles. And it's useful for us to be able to look at a certain number of genes and figure out which individuals have which alleles at those genes. And you can do a lot of stats and calculate things like genetic diversity from that. So the type of gene that we were looking at are called microsatellites. And so these are areas of DNA that are made up of repeated sequences. And different alleles of these genes have different numbers of repeats, so they are different lengths of a DNA fragment. And this is actually very useful because you don't have to do any sequencing to figure out which allele. All you have to do is look at the size of a DNA fragment. So this allows us to extract DNA from our fin clip samples and then use polymerase chain reaction, or PCR, which maybe some of you have heard of, which basically just isolates, concentrates the samples into just the genes that we're looking for. And then we can run them on an instrument called a capillary electrophoresis machine, basically, that separates DNA fragments by length. And so this separates the different length alleles from each other. And so we can tell which individuals have which allele. And it shows up, it's kind of hard to see here, but it shows up as peaks on a graph like this. So this is one individual that has two different alleles that are shown as these two peaks on the graph. So that's a heterozygous. And so from this type of data, we can calculate different measures of genetic diversity. And so that's what I wanted to do, to look at how genetic diversity changed from before to after fire. So I calculated 3 different measures. First of all was rarefied allelic richness, which is essentially just the number of different alleles that occur in a population. So if a lot of different alleles occur in a population, that's a reflection of high genetic diversity in the population. Unexpected heterozygosity is another common measure of genetic diversity that's used, and it can, homozygosity is the opposite of heterozygosity, where individuals have the same allele, and that can lead to bad things because basically it can lead to the expression of deleterious traits, and it's the manifestation of inbreeding, essentially. And then the third one is effective population size, which is it's mathematically calculated differently, but essentially it's a measure of how inbreeding is expected to affect a population. So if you have a low effective population size, that's a very sparsely genetically diverse population that will experience heavy inbreeding. And then the second type of analysis I did was looking at spatial genetic structure. So this is looking at this area right here. We're deciding whether or not there are distinct genetic groups throughout this, and how genetically similar are those groups, or are there like very isolated groups, or is it one genetically homogenized population? Now I'll move on to talking about the results here. So these are the changes in genetic diversity from before to after fire at burn sites versus unburned sites. And so you can see the 3 different measures here, and on the top is our unburned sites. And what those values mean, they're positive values, so it's above 0. That a positive change means that there was an increase in genetic diversity from before to after fire at unburned sites. But then if you look at the burn sites, from before to 1 year after and from before to 2 years after, we saw moderate decreases in genetic diversity. So this shows that burn sites and unburned sites had significantly different trends in genetic diversity, where there might be some kind of underlying trend that's causing an increase in genetic diversity at sites that are not affected by fire. But then the fire comes through, sites that are burned actually show a decrease, which is very different from the control trend. So this is evidence for significant effects of fire on genetic diversity in trout. So then our next analysis, looking at spatial genetic structure here. So I'll explain kind of how to interpret these figures. So on the left side there, we have the pre-fire data. This is within the Peter Headwaters Project reclamation area, and all of the pie charts on the map represent different sampling sites. And so the bars on the top there, if you look at the very thin vertical lines there, those are individual brook trout, and the colors represent the proportion of their genetic signature that is attributed to a certain genetic cluster. And so this analysis found evidence for 6 distinct genetic clusters that occur within the Poudre Headwaters Project area. And so that those red pie graphs there on the top left, that's a Corral Creek drainage. And so what we see is Corral Creek is very genetically isolated from the rest of the area, whereas the rest of the area kind of some slight genetic segregation based on major tributaries. But what it shows is that individuals are basically breeding within each of their own geographic areas. And so it's causing distinct genetic groups to form. But then when we look at right after fire, that structure is disrupted. So this is evidence that the fire is either causing mortality of fish and then recolonization of fish into certain areas from a different genetic cluster, and the combination of it's motivating fish to move. So it's motivating fish from a certain genetic cluster to migrate away from a severely burned area into unaffected habitats. And to look into that a little bit further, I did an additional analysis that uses genetics to look at directional migration rates. So through some fancy statistics, you can actually take a data set and estimate if it's likely that fish were moving more in one direction than in another direction. And what we found here is that fish were moving. It might be a little hard to see on this map, but those 2 top circles there are burned areas. And the directional migration rates showed higher migration from burned areas into this unburned region there. So those numbers are like how many times higher the migration was in that direction as opposed to the other direction. And so this kind of provides evidence that fish movement was initiated from severely burned areas into unaffected habitats. So to summarize these findings, we saw decreased genetic diversity after fire at burn sites, which is evidence of population bottlenecks that decrease genetic diversity. And we also saw disruption of the population structure within the Poudre Headwaters Project area, where spatially segregated genetic groups were mixing together. And this was probably because fish movement was instigated by the fire from burned areas into less affected areas. So I think these findings have some pretty significant implications for native trout management. So we found that fire can reduce genetic diversity and motivate trout movement away from disturbance events. So these types of impacts could be more severe for restored native populations. So what we're studying here are brook trout, which are super abundant. They don't suffer from low genetic diversity concerns. They often exist in habitats that are highly interconnected, and there's a lot of access to other populations to recolonize when locally certain populations might become extinct. But this is not the case of trout like greenback cutthroat trout. So they have lower baseline genetic diversity. They often exist in small and isolated habitat patches where reclamation projects have occurred. And so there's lower potential for recolonization after a disturbance event like this. And so I think this shows that larger reclamation areas, such as the Poudre Headwaters Project area here, might improve native trout resilience to disturbance events in terms of genetic diversity. So what you can see here is this large reclamation area has part of it that was burned, but it's so large that a subset of that remained unburned, and it allowed these brook trout to basically flee the disturbance and have a habitat refuge in the unburned portion. And also it allows individuals to recolonize, individuals from the unaffected areas to recolonize, even if local extirpation does occur in the severely affected portion of the habitat. So I want to thank all the organizations and sponsors of this project and the Cutthroat Trout chapter here for the generous scholarship. That's a huge help. And all of my field techs who volunteer their time to help collect these genetic samples, and all of my collaborators. And I'll open it up to questions.
costly, if the wheel better to go. I'm sure it Trout may be more adaptable to surviving versus another trout in a different league.
That's a good question. So some work that has been done has showed that larger fish are much more resilient. So they're less likely to be killed and they're more likely to withstand these types of things. In terms of genetic factors that might make individuals more resilient or more susceptible, that's a great question. I think some cool research could be done, but these genetic markers that I was using aren't very informative about that type of thing because they don't actually code for any traits. They're what we call neutral markers. So it's DNA that just exists and doesn't really do anything that we know of. And so it doesn't manifest as like differences in morphology and things like that. But it's useful for looking at genetic similarities between populations and like overall levels of genetic diversity. Right. Yeah. So the question was, how did we get before and after fire data when we didn't know where the burn was going to happen? And the answer is that it was just pretty much lucky. I mean, it's not lucky that the fire happened, but for our purposes, we happened to have an assortment of multi-year sampling sites that had been sampled year after year, and then the fire came through and burned about half of them. And so it manifested as a fortunate opportunity for looking at the effects of fire on trout. Yeah, definitely. So the insight is that the more options or the more interconnected habitat that trout have access to, that's going to lead to greater resilience to things like fire because they have access to unaffected habitats and also have genetic connectivity to other populations that might not be affected, as opposed to a really small population like maybe some of these reclaimed greenback populations that exist in like 3 miles of a single stream. Fire could easily wipe that entire population out, and there's no potential for recolonization because they're isolated by barriers. That's a good question. I'm trying to figure that out. Well, I think I finished up my master's in December, and after that, I'm going to try to apply for jobs. I think like an agency biologist position with something like Parks and Wildlife or the Forest Service, something like that would be great. Thank you. All right.
Yeah, thanks, Tyler. It was pretty devastating to watch that fire grow and burn that, and we're glad we hopefully will give Colorado Parks and Wildlife some information on how to work restoration of those areas, and also the Forest Service about you know they like to see natural burns, but that was a little bit out of control. So we'll see if there's a change in any philosophy there. But again, thank you so much for your insights there. We appreciate that. And good luck to you after your studies. And hopefully we'll see you again as a somebody being called upon to help restore some other streams and everything like that. So again, all right. Well, I guess that takes us to the end of our presentations, and we could have the raffle drawings.